Dr. Hugo Volkaert. He had a bachelor and a master's degree in agricultural engineering in Belgium and got his PhD in forest resources from the University of Maine, the United States of America. And now he is working with the Center for Agricultural Biotechnology in Kasetsart University, Thailand. His research of interest are the population genetic diversity and phylogeography of forest trees, genetic diversity and domestication of bananas, and also in some economic plants such as the oil palm. Not only the diversity study, he also used biotechnology as tools for crop improvement. Today, he will talk in a topic of application of genomic tools to plant germplasm characterization. Please welcome Dr. Hugo.
Okay. Hello. Okay, thank you for the introduction. Thank you for inviting me. I changed the title a little bit. Genome skimming is one technology of genome genetic characterization. Is the sound okay? Screen sharing okay? Everything?
Yes, everything's okay. Fine.
Okay. Unfortunately, we're living in a world that is in crisis. And the future doesn't look too bright always. We have a growing human population, and that worries us because we have to feed those people. There's an increased use of fossil energies. That's one of the consequences is carbon dioxide released and climate change. But closer to our hearts is the loss of fertile agricultural soils, salinization, erosion, and unfortunately, there are still world leaders around that prefer weapons over well-being of people. We have to deal with all those problems. Now, around 2008, some big chemical agricultural seed companies came out with climate-ready crop seeds. And essentially, they are genetically engineered crops that can withstand, that supposedly can withstand drought better than regular crops. But do we really need climate-ready crops now? So at that time, there was some discussion, and I just thought, and I'm still convinced it's just a marketing gimmick. Yes, they are a little bit drought resistant, but we don't need climate-ready crops. What we need is climate-ready breeding, climate-ready breeding programs, climate-ready germplasm collections. We don't even know what the climate will be like in 10 years, 20 years from now. Even though, yes, we may say it may get warmer a bit, may get drier a bit, but where exactly, how exactly, we don't know yet. Unfortunately, we cannot wait until it happens and then start preparing to remedy it. So we have to prepare now so that once we know how the new environment will look like, both physical temperature, precipitation, and biological, new pests and diseases. So as soon as we know what are problems at hand, we should be able to adapt crops. We should be doing breeding within a few years to give better crops, better adapted to the new environment, to the farmers. And what do we need to know? What's the knowledge we need for a climate-ready breeding program? We need access to germplasm. And as Ms. Kanyapon and Dr. Watanabe have talked about that, we still don't know enough about germplasm, and we still have not collected enough, and definitely we still have not protected enough, especially of the wild relatives. We need to have access to genetic information, and that is what I will be talking about mostly today. But we also need to have access to phenotype information And that's why I asked the question to Ms. Kanyaporn, in how many environments are the germplasm collections being tested when they're grown out? Basically, we need adaptation information. We need to know how a crop would adapt to new physical environments, temperature, precipitation, or biological environment, pests and diseases. And germplasm genotyping can set us on the way to do that. Germplasm genotyping started already many years ago. Let me call them first-generation molecular markers, restriction fragment length polymorphisms, and then came rapids and AFLPs. After that came markers like simple sequence repeats or microsatellites, and all kinds of derived similar markers, simple sequence repetitive regions, or inter-retrotransposon markers and so on. But since the beginning of this century, the technology is moving very much into the sequencing-based analysis of genotypes. So that's what I will be talking about. So why do we want to do sequencing? Because there's probably much less homoplasy in sequence-based information than there is in microsatellites. Microsatellites, you can have a fragment of exactly the same length, but that does not mean by no means that those two fragments are actually related to each other. They may have a different evolutionary history. With sequencing, we can get good information on the diversity, but also on linkage or linkage disequilibrium of markers. We can get good measures of population structure and population size. We can study what part of the genome is shared among accessions, the core genome, and what part is the additional, and that combines then total for the so-called pan-genome. Sequence-based analysis is also immediately accessible for genome-wide association studies, and that way can detect particular loci that are involved in adaptation to the environment. I have to admit, as of now, sequencing-based analysis of large collections is not feasible yet, not affordable yet. For some of the crops or the plants that Dr. Watanabe showed us, they are of local importance at the moment. We have no knowledge about their genetic backgrounds, and it's still too expensive to do a sequencing of a few hundred of those accessions. But in the next 5 to 10 years, the cost will come down sufficiently that we can actually do that. So one of the methods that a certain group of people advocate is called genome skinning. Just barely touching the genome, let's say, just having a little read-through of the genome to get the most basic essential information. However, the way it is being done mostly at the moment, I would rather call it genome skimping rather than genome skimming. Is it better now? Sorry, I forgot about that. What is genome skimming? Well, it's essentially doing whole genome sequencing at a very small scale, low coverage so-called, essentially barely 1x to 2x the genome size. So if a genome is 500 megabase for a typical plant genome, up to 800, and you ask for a sequencing output of 1 gigabyte, you have about 1x genome coverage. Why do people use it at the moment is to get basic information, mostly on repetitive DNA sequences. In theory, you could get the whole genome, but since it is a random sequencing protocol they use, some parts of the genome are sequenced more than others, and many parts may be missing. So, but the repetitive DNA sequences will be sequenced almost by default. So in plants, the chloroplast, and in animals, the mitochondrion, also the ribosomal loci will be sequenced. And that's very useful for a very brief study of taxonomy, grouping, and so on. You can develop DNA barcodes from that, but in my opinion, it's not sufficient. You have to do a little bit more. Just a few months ago, I think, about a month ago, was this article published in Nature about the chickpea genome variation. And there, for chickpea, it's an important crop, especially in South India and Africa, Southern Asia and Africa. And so they did more than just genome skimming. They got some more data, analyzed 3,300 something accessions, and then compared them and combined that into a so-called dispensable genome, which are parts of the genome that is found in just one or a few accessions, and the core genome, which is the essential part of the genome, and then that total adds up to the pan genome, which is all the genes and sequences present not in one individual, but in one species. And the core genome is the set of genes and sequences that are conserved among all those accessions. And the dispensable genome is then the rest, so the part of the genome that is being found only in one or few accessions, not in all of them. And here you see a comparison of the diversity, and there's a blue, pinkish, reddish, green, and purplish brown set of circles here. Each time the outer circle, which is lighter colored, is the diversity in the cultivars, while the darker circle is the diversity in the wild accessions. And that is for SNPs, so nucleotide substitutions in bluish colors there, for insertions in reddish colors, and for deletions in these greenish colors, and for transpositions in these purplish brown colors. But you see, each time the cultivars have much less diversity than the wild accessions. So this is the kind of study that can prepare you for a breeding program that is ready for climate change. You have all the genetic information at hand, and if you can combine that with some phenotype information, you're set to go. But that is a very expensive effort, so you need a bit more data than simple genome skimming. I have been doing similar kinds of research on small scale on forest trees and bananas, and I try to get about 10 times genome coverage rather than 1 time. But I still do that with a minimal investment. My budgets are very small. So what do? Is I talk to service providers and I asked them for their most efficient sequencing protocols, where you get most DNA information output at the lowest possible cost. And then they recommend me then just to follow the resequencing protocols that they use for human genome sequencing. What I get is one library that gets sequenced in a single run. The only thing I negotiate with them is that I want the insert size of the library to be as long as possible without additional cost in either money or in quality of the output. I read in the literature, and I've heard from some people that they can sequence libraries with DNA fragments up to 800 base pair long. But when I try to get commercial providers to do that for me, they are very reluctant because the quality of the DNA sequencing output will not be as good. And so then they have to do more sequencing and they will charge me more money. So I have to settle mostly for about 500 base pair insert library. Even though I wish it could be a little bit longer. But at the moment, my inserts, my fragments are about 500 base pair. What is a typical output that I get? About 12 to 15, 16 gigabytes of DNA sequence data. I asked for pair end and 150 base pair reads at either end. So my fragments are 500 base pair, 550 base pair, and I get 150 base pair on each end, so total 300 base pair. And the central part of my fragment is unknown. With this approach, I get the quality to be quite high. More than 85% passes the quality level 30, which is 1 in 1,000 error. 93%, that's the lowest I have, passes the Q20 quality output. So that's 1 error or 1% error. Total of N nucleotides that I receive from them is less than 0.01%, and very few reads have more than one single N ambiguous nucleotide. I do get some duplicate reads, about 5% at most. What are the outcomes? Well, like typical genome skinning output, I do get information on the chloroplasts. So this is for 2 forestries here in Thailand. That is not difficult. I also get complete mitochondrial sequences, though not always in one circular molecule. Okay. For technical reasons, because Illumina fragments of 500 base pair may not cover the long repeats, but also for biological reasons, there is no biological guarantee that the mitochondrial genome is a circle. What else do I do? I spent quite some time analyzing the K-Mer profile. From that, I can learn about the genome size, heterozygosity. I can learn about the repetitive part of the genome. I will briefly explain what is a K-Mer. If you have a sequence, so either from a genome or, in my case, from a sequencing reads 150 base pair or 100 base pair long, you can chop that up in shorter fragments, and that are the K-Mers, the K nucleotides. For instance, in this case, I chose a K-Mer of length 27, and over a 100 base pair long fragment, you can move that from the beginning to the end, and you have 74 different K-Mers. So here's an example of sliding it across sequencing. And what you do then is you count this particular K-Mer sequence. How often do you find that in your data set? Either your sequencing reads or your genome. And in case of a polymorphism in your biological sample, let's say a diploid individual where you extracted DNA from, then for a particular position, you get 2 different K-Mers. Each of those 2 K-Mers you can expect them at half the frequency, because one comes from one chromosome, the other comes from the other chromosome. But one SNP here introduces 27 new different K-Mers at the last position, the second last, and so on. But when you slide the K-Mer across the SNP position, you have 27 different K-Mers. So the result is that polymorphic size have double the K-Mer types, each of them at half the frequency. And if you make your K-Mers longer, from 27 to 31 to 45, then more of the K-Mers will cover a polymorphic site, of course. And that results in what they call a K-Mer spectrum. This is a typical example from the website of one of the computer programs I use, so the K-Mer analysis toolbox. And actually a spectrum like this is composed of many different parts. There are one group of sequences that are very low frequency, but there are very many of them, and these are the sequencing errors. Then there's a group of sequences that occur at single occurrence, so I would call them heterozygous. Then there are sequences that occur at double occurrence, twice the amount, so this is 20 times you find it, this is about 40 times. So these are the double occurrences, could be the homozygous. And then you have triple and quadruple and more occurrence. These are the repetitive sequences. So combined, these distributions make up a skewed global distribution of the whole genome. Plus here a peak or an indication of the sequencing errors. So I use the program KAT, K-Mer Analysis Toolkit, for that mostly, but also some others. And here in blue is, for instance, the K-Mer profile I obtained for a coconut genome. And that is split into a sequencing error part. There is here a green peak of the single copy or single occurrence K-Mers, and a red peak here for the double occurrence K-Mers. If you combine the green and the red, you get this orange spectrum here, and that is almost a complete set of the K-Mer spectrum, except here at the beginning there are the sequencing errors, and here at the end, the higher frequency K-Mers, these are the repetitive K-Mers. And using these programs, you can actually separate them more or less. Many people have done that, but mostly that's reported only for a single K-Mer. I do that for multiple K-Mers, and then you get very nice pictures. I'll explain them later, but this is what you get for Arabidopsis. I downloaded a data set for Arabidopsis genome sequencing from GeneBank. And so you get your errors at the low frequency end here, and you get a K-Mer spectrum. And no matter how long you choose your K-Mers, there are very similar spectra. Here I did that for a human sample, and it almost looks like Arabidopsis. This is from a Korean individual, downloaded from GeneBank. It almost looks like Arabidopsis, except there is a little bump here in my distribution. And you see this is about 15, while the big peaks is about 30. So this little bump here are the polymorphisms, the heterozygosity. That's in human. Here this is in teak, so a forest tree. And you can clearly see there is a definite bump. This is the 1X occurrence K-Mers. These are the 2X K-Mers. And by comparing the area under the graph, you can estimate the heterozygosity of a sample. And here I indicated this is for another forest tree, Xylea. These are extremely heterozygous trees. You see at very short K-Mers, you see 2 bumps. The 1X heterozygous, 2X homozygous parts. But as soon as your K-Mer becomes a little bit longer, you actually see only a single peak. But this is not a single peak for the homozygous part. This is the single peak for the heterozygous part. So these trees have highly heterozygous genomes. So I've shown you the example of coconut before. So this is this sample I showed before. You can separate into 1X and 2X parts. I did that for multiple K-Mer lengths. This is in a tall coconut phenotype. This is in a dwarf coconut phenotype. And that has been known from many other studies that the dwarf coconuts are highly inbred homozygous accessions, while the tall phenotype is outcrossing and consequently heterozygous phenotypes. I did this also recently for a cocoa accession I downloaded from GeneBank. And you see cocoa is moderately diverse, moderately heterozygous. Here from banana. I do study bananas and I try to find the origin of the cultivated bananas. And here is K-Mer analysis for some different species. The top one here I collected myself in northern Thailand. And the bottom is downloaded sequence data from GeneBank. But we found a population in northern Thailand in the forests that is triploid. And it took me some time to figure out exactly what is going on. But now I can clearly, confidently tell you it's a triploid AYY genome. The A is for Musa acuminata, one of the species that is also in edible bananas. And the Y is for Musa yunanensis. I did find one diploid individual so far in northern Thailand. And when I sequenced this, so then the chloroplast, mitochondria, ribosomal sequences clearly pointed to Musa yunanensis affiliation. And so the hybrids, triploids, have one acuminata genome and 2 yunanensis genomes. And that gives this very strange distribution. But you can see here a peak at 1X, a bump here at 2X. And for the shortest K-Mers, you can see there is some kind of a distribution here at 3X. It fits nicely, 1X, 2X, 3X. So these are the YY genomes. And of course, everything that is acuminata comes in single copy and is different enough from the yunanensis genome that it ends up here. And the yunanensis genome itself is highly heterozygous. If you compare that to Musa acuminata subspecies genomes that I downloaded from GeneBank, Acuminata banksii from southern and eastern Indonesia Papua New Guinea, it's almost homozygous, and that's known. Zebrina from central Indonesia is more polymorphic, but Germanica from Burma, Thailand is even more polymorphic. Not as polymorphic as some other forestries, but quite enough to cause some problems for the analysis. And then the big surprise was this here. This is a cultivar of bananas from Thailand, New Munang. It's a triploid. We knew that. And we thought it might be A and B, yes, but I had done some analysis before and it was very strange. So we did a whole genome sequencing analysis plus a flow cytometry analysis to confirm it's a triploid. And the K-Mer spectrum here looks almost like a homozygous individual, like Arabidopsis almost. Notice that my genome, my coverage is much lower than some of the examples I've shown before, like the Arabidopsis I downloaded, but that's on purpose. I cannot spend the money to have it sequenced at much higher coverage. But turns out from the sequences, I can find it is an Acuminata genome in there, a Balbisiana genome in there, and Musa itinerans genome. These are 3 different genomes. Don't ask me how nature did this, but there seems to be something very strange here going on. An A genome combined with a B genome combined with a itinerans genome. Still have to figure out some more details how this came about, but it's very interesting. And this I can confirm through low coverage genome sequencing. Here are some examples of different tree species, all in the Dipterocarp family. 3 Dipterocarpa species and 1 Shorea species. And so this is genome coverage at 5 to 10x. But in these examples, I can conclude that Shorea, and I have 2 other Shorea species, and they are similar to this. Shorea is less heterozygous than Dipterocarpus, at least for the species grown in the dry forest in Thailand. Here I do this in a forestry species in 1 single species, 6 different individuals collected in Thailand. And you can see that within this 1 species in Thailand, there is diversity in the level of heterozygosity from almost 2% in this individual to about 1.25% in this individual. These are rough estimates. These are not exact figures, so don't. It looks nice. I say that 25.2, but don't believe that exact number. But it ranges from about 2% to 1.3%. So there is quite diversity within Thailand, just in the level of heterozygosity. So what did I get from this K-Mer analysis? I can estimate the genome length. I have not talked much about that, but my analysis confirms for banana it's about 500 megabase. For the Dipterocarps, it is 350 megabase, and for Shorea, it's only about 300 megabase. I can get information about the repeat content. You have to look at the tail of the distribution, and I can get this very interesting information about heterozygosity. Heterozygosity is interesting because it tells us about the biology of the species, whether it's outcrossing or inbreeding, and how much outcrossing there would be. But with the low coverage genome sequencing I do, I also get information about the genome assembly. I can assemble the genomes, fragments, of course. It's not a telomere to telomere complete chromosomal assembly, far from that, but I do get fragments that are more than 100,000 base pair in length. For all my genomes that I have assembled so far, there are each time 100 to 200 up to more than 1,000 fragments that are more than 100,000 base pair long. The longest fragments I get are from Shorea species, and they are up to 1.2 million base pair long. So these are substantial fragments, good enough to detect genes, single copy genes. If you want microsatellites for analysis, you have them. So you get the gene sequences and you get the other information. Here I analyzed those and searched for how many of the so-called essential genes, single copy genes, can I find back. So BUSCO analysis. And you see here 75 to 85% of the so-called single copy essential genes that one should find in all plant species, I get them, single copy. 5 to 10%, I find them duplicated. Now, there are two reasons why I might find a duplicated sequence here. One is that because of the high heterozygosity, some parts of the genome are assembled as different chromosomes, different fragments, even though they are actually the same locus. But I did some further analysis, and it turns out that all the individuals here with lots of orange or lots of duplicate genes belong to the diptercarps and to gluta. And these are true gene duplications, I think. I did not check for every gene, but for several genes, I do find 2 copies in the diptercarps or in gluta. And Anacardiaceae, a relative of mango. Like leafy, which is in almost every plant species a single copy gene, I find 2 copies of that in diptercarps. There are also some parts of the genome of the genes that are fragmented, and yes, there are some missing. But by and large, I get decent information about the genes, and more than you need actually for a genome analysis. What's more, even with that one single library that I sequence at intermediate coverage, let's say, I can not just assemble genome fragments, I can actually combine them into haplotypes. That's because of the high heterozygosity I find. There's so much heterozygosity that even with the Illumina sequencing I do, I find multiple SNPs, multiple polymorphisms within a single read or within the 2 pairs of reads that belong together to the same fragment. And I can, in many cases, reconstruct gene-spanning haplotypes that are 10, 15,000, 20,000 base pair long. So get the 2 chromosomes and all the SNPs on them all along over a region, more than 10,000 base pairs. Why do I think haplotyping is important? Because linking of the SNPs into longer alleles gives you additional information about the ancestral descendant relationships. I'll show you just in a minute. With haplotypes, you can get much more robust information about population structure. And that's an important part of what we are trying to find out. Structure in a population, structure in a species, structure in the germplasm you collected. Is that geographically structured? Is that environmentally structured? Or in other ways? And so that helps to establish causal relationships between your haplotype and the phenotype. So this is an example we did about 10 years ago, starting about 10 years ago, so published 6 years ago now, we amplified a particular gene, isosaturated dehydrogenase, from these 3 Xylia. We collected 540 individuals, so which is 1,100 chromosomes about. The fragment is only 686 base pairs long. Within that short fragment, we find 34 nucleotide substitutions and 7 insertion deletion polymorphisms. They combine into 36 haplotypes. So these are the network connections for those haplotypes. Each line here represents a mutation, a polymorphism, either a base substitution or an insertion or deletion. And the size of the circle corresponds to the frequency of this particular haplotype in the total population. So the very small ellipses there, these are single copy occurrences of this particular haplotype. The larger ones are 2, 3, 4, 10, 15, 20 times, and several hundred times. So we have a total of 1,094 chromosomes that we analyzed. They're all represented here. And this way we can clearly see what are the common haplotypes and how are they related to each other, and what are the strange haplotypes, like this one here. This is 3 polymorphisms different from this common one. This is a population structure analysis for these 547 individuals. And we don't see that much structure geographical structure within Thailand, but in the western part of Thailand, these are all from the western part of Thailand. Green is the northeastern part of Thailand, and red is the northern part of Thailand. So here, red and green is north. This is western, northwestern here actually. So we can link the geographical distribution with the population genetic diversity that way. Here I show you similar kind of analysis for bananas, wild bananas, Musa acuminata only. So with this kind of analysis, we can separate the different species. Many banana cultivars are hybrids between acuminata and balbisiana. So this is only the acuminata. So for the hybrids, there are among here, only the acuminata genome is analyzed here. Balbisiana genome is separately. And we can distinguish several clusters, and some of them correspond nicely to a subspecies. Some of the clusters are closely linked to each other. Truncata is very distinct from everything else. Here, this is Halabanensis from Sumatra. It's very distinct. This is subspecies Sumatrana. This is the central core of Zebrina, Malaxensis, Banksii, and Burmanicoides siamea. And this is Pahang, which is the reference genome is Malaxensis. These are the other Malaxensis, so it's not a typical one. Banksii is nicely distinguished. And then there are these things here. These are 3 individuals, 3 haplotypes I found in Thai cultivars. I have no clue, no idea what they really are. And these are 3 strange haplotypes we found in Sumatra wild bananas. Morphologically, they don't look very special, but genetically, there is something going on there. Another thing we find out here is that these 5 open circles here These are haplotypes we found in cultivars, but up to now have not found in any wild relative. Not in Thailand, not in Sumatra, not in some of the other places I have access to. So these are, and mind you, these are the haplotypes found in the very common Cavendish, Gros Michel, and Mysore, Pohm, and these are the haplotypes found in the most important cultivars. We don't know where they come from in the wild. This is really the power of haplotype analysis. What else can I do is using coalescent-based analysis based on either the linkage disequilibrium or the so-called side frequency spectrum analysis. Side frequency spectrum is actually the distribution of SNPs among accessions. Either type of analysis can be done in a coalescent framework to estimate past population size. How many individuals were around 5,000 years ago, 10,000 years ago? This is done quite often for human populations. I will show you some examples where I've done this for plants. The only problem I have for both bananas and forest trees is what is the generation time? Because actually this analysis is based on generation times, and for humans we have some idea what is an appropriate generation time. For bananas and forest trees we actually have no clue what would be the generation time. So this is the analysis for a wild banana from northern Thailand, Musa acuminata, morphologically belonging to Malaxensis. And you see there has been a gradual decline in population size. Don't look at this here. This is an artifact of the analysis. And don't look here at the years, because this is assuming a generation time of 25 years. Mutation rate is more or less appropriate, but generation time, we have no idea what is the generation time of a wild banana. We know they're perennial, they can live for many years, but what is the average time, number of years between parents and offspring? Effective population size in 10,000, so this is 10,000 up to 50,000, 60,000 here. This is for bananas. This is for some of these forest trees. You get different types of patterns. They have gone through some cycles. And again, don't trust the years here. For forest trees, we have even less idea what the generation time would be. And this is an example of this kind of analysis for teak in Thailand and from Eastern India. And we see in Thailand, for most samples here, 4 different individuals, a clear decline in population size. Here, this particular individual has some increase, and that is also found in Eastern India. This is probably a climate change related, past climate change then, not future climate change. It indicates that the climate has changed to some extent, and that in Thailand we are more at the edge of the species distribution, and so they have suffered a little bit more, while Eastern India is closer to the core of the distribution. There is some more diversity there. So why do I do this analysis? Because I want to know about the current status of diversity, whether it's in forest trees or in crop germplasm. I want to estimate parameters of current diversity dynamics. and I want to estimate the dynamics of the past, combined with some phenotype and environmental data that can lead me to meaningful conclusions about adaptation. How did these plants adapt to different environments in the past? That because of their past and present population diversity and breeding system. If we can derive some conclusions about the past, then maybe we can have some reasonable predictions for future climate change adaptation. However, whole genome sequencing is still too expensive at the moment to do it for many individuals. So my trees, 5 individuals, 2 individuals per species, that's about all I can afford. Last year, August, I paid $350 per sample. I expect within the next year or two that will come down to $200 or $150. Illumina is going to introduce some new sequencing kits that are more efficient and lower cost. But at the moment, for the next 5 to 10 years, probably we have to do some kind of reduced genome analysis, genotyping by sequencing, GBS, sorry for the error here, or RAD sequencing, restriction-associated DNA sequencing, DART sequencing. These are all techniques that let you sequence some part of the genome at sufficient coverage to do proper analysis. But I would actually advocate, rather than these random approaches, to specific capture particular regions of the genome for sequencing. There are, at the moment, some chip-based technologies, but maybe we should go for PCR-based technologies where we use primers to amplify particular genes in a genome. For instance, here, this is Gigantea gene, which is involved in photoperiodism. If you compare the genes of different plant species, flowering plants, monocots, dicots, but also non-flowering plants, you see there are some regions that are extremely conserved. In all plants, this particular region is conserved. I can make a PCR primer for this part and another conserved region of this gene and amplify it. And that is what I get here. This is Gigantea amplified in sugarcane, teak, a dipterocarp tree, rubber tree, and a fern. In almost all of these species, I can see one band, or dipterocarps, 2 bands, as I said, lots of duplicated genes. Even in ferns, I can see some appearance of bands. This is here another gene, a small gene family, phosphate transporters. In all species, I can amplify the gene. This gene does not have introns in most plants, so they are all about the same length. This is isocitrate lyase, again in a fern, rubber tree, dipterocarps, teak, sugarcane. We can amplify these fragments. So if we do this for a number of genes, I have a set of, I think, 35 primer pairs now. We could amplify the same genes in all species and sequence them at high coverage. They contain gene sequences, but also introns. And so we get sufficient information for good population genetic analysis and good diversity analysis. So in summary, with my whole genome sequencing, I use a single library shotgun approach. and that gives me very useful information at an affordable cost for small-scale experiments. I do spend about double the amount of money what genome skinning researchers spend, but I get about 20 times the amount of data. So for a little bit more cost, I get much more data. Why? Because the library construction is almost fixed cost. Whether you get 1 gigabyte output or 15 gigabyte DNA sequencing output, you have to construct the same library, and the cost is probably within a few percent the same. You can save a little bit on volume of particular enzymes, but not much. So I get about 10 times the DNA data from the genome skimming approach, and I estimate I get maybe 200 times the information, because I get additional heterozygosity data, I get phylogenetically informative nuclear markers, I get markers for parentage analysis, and that's more important in forestries than maybe in germplasm collections, but for wild relatives it may be informative. And I can do population genetic analysis for the present populations and the past. In addition, I get comparable data across different species. So I can do the same analysis in bananas in my forestries, in your roselle, hibiscus accessions, or your ginger accessions, or your rice. That is one of the major advantages of this approach. If people are interested in doing this, I would recommend that you do sequencing with longer fragments, as long as you can get without increasing the cost too much. So, but companies can do it at 500 base pair fragment size. Do get paired end sequencing. It is worth the effort. 100 or 150 nucleotide reads is optimal in my experience. I have tried 250, thinking you get more information, but actually the quality of your information is not that good anymore. And insist on high quality reads. Don't let anyone send you low quality results. To be honest, I showed you the example of cocoa before, the K-Mer analysis I did. I downloaded from GeneBank, and I have to say that the quality of that sequencing set is very bad. 25% of the output has to be discarded because of repeats of sequencing. So they did PCR amplification, and another 15% has to be discarded because of sequencing errors. When you do the quality analysis with FastQC or other programs, I was amazed that the quality is extremely high. So the read quality is extremely high, but actually it is artifacts. It's artifacts probably from the PCR reactions during library construction they used. When I did a K-Mer analysis, I clearly saw that there's an extremely large amount of single-ton or double-ton K-Mers. So with that, I would like to acknowledge the Center for Agricultural Biotechnology and AgBioPerdo program for supporting some of the sequencing and analysis of the research I did. Some of the students that have been working on this for the past 20 years on different aspects of this research, and I would like to thank you for your attention. I'm happy to answer your questions.
Thank you, Dr. Hugo. If you have your germplasm collection ready, you can do same thing that Dr. Hugo did. So if you have any questions and you want him to help, you can contact him at any time. Anybody have the questions? I think. Okay, that's questions from the chat box.
Okay.
You want me to read it, or?
I'm reading it, but you can read it aloud for the other listeners.
Okay. It's from Ms. Lear. She said that it's very interesting. Congratulations, Hugo. You have newly discovered that musa uanesis. Okay, I'll go to the end. Okay. The question is it the addition to previously recognized ancestral genomes in banana, ABST, has thus information been published? Okay, you go for the.
I'm writing up the manuscript. So this is really very new. I would say just off the press. I hope to have it published within this year. But yes, it is the first time that we find an itinerance genome in any cultivar. And I can tell you, I haven't done the sequencing yet, but we are preparing the morphological analysis and so on. There are other hybrids of Musa acuminata out there in the forest. So not only itinerans or yunanensis that process with acuminata in the wild, there is also rubra, Musa rubra, that is crossing with acuminata. I don't have the sequencing data yet, but okay. What type of raw material sequences and tools to generate those rainbow curve figures? Well, this is from whole genome sequencing. So what we do is I extract the genome from the plants, and I try to get rid of chloroplast and mitochondria, but it's still sufficiently in there to analyze them. And then, so we do the sequencing. We receive the reads information back from the service provider, and then we chop that up in those K-Mers. So you have a sequence of 150 base pair that you receive from the company. We chop that up in 27 nucleotide long stretches. We let the computer read all those K-Mers and cluster them, compare them, and then count for this each particular K-Mer, how many times does the computer find it? Once, that's probably a sequencing read error. Twice, dubious. Do we find it 5 times? That particular sequencing read? Do you find it 15 times or 25 times? So the computer makes a very big Excel sheet for you. For each K-Mer, how often does it find it? Then you summarize that and you say, okay, how many K-Mers were there that you find once? So the computer will make a second Excel sheet and say, okay, K-Mer occurrence once, you find that so many different K-Mers. K-Mer occurrence twice, you find it so many different times. Then let me try to get back to the slides here. Sorry, now I stopped sharing. We have to share this here. Sorry. And let me go back to one of those rainbow figures. Okay. So you ask the computer how many K-Mers were there, you find ones. That are millions. Okay. This is 1 million, excuse me, 1 million, 10 million, 50 million times. And 50 million times you find a K-Mer, a unique one. How many times do you find a K-Mer? 5. How many K-Mers are there that you find 5 times? There are some. 2 million, 3 million. How many K-Mers are there you find 10 times? Okay, here, about 1 million. How many K-Mers are there you find 49 times? About 7 million. So that's for this particular K-Mer length, 45 base pair long, you get a distribution like that. These are sequencing errors. These at the tail here are the repetitive sequences. And this area here is the single copy, mostly gene coding region. Okay. Here, show you the cocoa example. You see here the peak of occurrence here is about 10 million here, but the peak of the errors here is huge. If you want to see the peak of the errors, you get a flat line here. It's several hundred million. So that's why I said this is bad sequencing. But the principle is the same. You count for a K-Mer of 17 base pair long, each different K-Mer in your data set. How many times do you find it? And then for each K-Mer that you find 10 times or 20 times, how many different ones are there? This is from an Excel sheet or another spreadsheet program, and then you ask for the graph. And then the rainbow is just because I colored them that. So all you need is your sequencing data and the K-Mer analysis program, which is essentially jellyfish incorporated in the K-Mer analysis toolkit, and then an Excel sheet, a spreadsheet. Now,
Dr. Hugo,
yes.
I have one more question. If for the breeding side, if I have the K-Mer data, I have haplotype, how can I use the data to decide for my breeding program? Like if I have germplasm, which plant should I pick to be parents to cross to get the good yield or whatever traits that I'm looking for?
Okay, so if you have the data, the sequence data, and you can reconstruct the haplotypes, you can reconstruct the relationships among these haplotypes like this.
Okay.
Now, you see there are, in this case, clear groupings. These are linked to different regions. This is Sumatra. This is Papua New Guinea. This is Malaysia Peninsula and Thailand, Peninsular Thailand. So there are regional groupings, but these are also morphological groupings, because these correspond to subspecies and they are morphologically distinct.
Yes.
Now, they can also group because of environment. they could also group because this could be domesticated and these could be wild. So there is no one answer for this, but based on this kind of analysis, this gives you a very great insight into the diversity of the species and some of the origins. Now, if you want to make crosses, sometimes you want to make crosses to maintain a particular type you have. So then you know you can make crosses within this group, for instance. And if, let's say, here, these circles here are in cultivars, but these darker colored circles are in wild accessions. So now I know these wild accessions are different from anything in the cultivar. So if I cross these, a wild with a cultivar, maybe I can introduce some genes from the wild without changing the genome too much, without changing the morphology or the yield or whatever too much. So this way you can decide, I want to make some close crosses, which ones would I do? On the other hand, sometimes you might make very wide crosses because you are frustrated. These cultivars are all susceptible to a particular disease or to drought. I want to improve that, and my best bet is to improve it by making a wide cross. Okay, you have different alternative wide crosses here. You know, doesn't make a much difference if I use these different. I can take one of these, representative of the whole group, cross it with this one of these here, cross it with this one of this cluster here, cross it, so you can make different wide crosses and see if that gives you the desirable outcome that you're looking for. So this is the kind of information you can get from this haplotype evolutionary analysis of genes. You cannot do this with microsatellites. You don't need whole genome. This is just one gene, one fragment. Okay, in banana, about 1,100 base pair long fragment. If you make it longer, you get more diversity, but that's not really necessary. This is an appropriate picture. I think you see clustering, you see that there is geographical diversity in there, there's morphological diversity in there. You can use that as a guide to make your decisions on what crosses to make or what crosses not to make, or why some crosses cause you problems. You may have been crossing these two different types and you always get very seeded. It's very difficult to germinate, or you get few seeds or whatever. Now you know it's because they are a very wide cross and maybe too wide. So.
Okay, thank you.